Assalamualaikum warahmatullahi wabarakatuh. How are you today? I hope my miss and my friends are good. Amin. My name is Muhammad Shahibul Alan, and now me and my group will be explaining about good laboratory practice, referring to the regulation of the Minister of Health under the Republic of Indonesia number 43 of 2013 concerning good laboratory practice is the implementation of activities to improve and strengthen the quality of laboratory examination results. The purpose of GLP is to regulate the way of running a good clinical laboratory so that it can provide quality service and results and can be accounted for. Clinical or medical laboratories must be properly organized by meeting organizational criteria, space and facilities, equipment, materials, specimen, examination methods, quality, safety, recording, and reporting. K3 Laboratory Officer or Team. The laboratory worker or K3 team has the obligation to plan and monitor the K3 implementation that has been carried out by each laboratory officer, including a. Conduct periodic inspection and directions on methods or procedures and their implementation, consumables and work equipment including for research activities. B. Ensure our laboratory personnel understand and can avoid the danger of infection. C. Investigate all accidents in the laboratory that allow the release or leak or spread of infection material. D. Supervise and ensure all documentation measures that have been taken in the event of a spill or splash of an infection material. A. Ensure that disinfection measures and are taken on laboratory equipment to be serviced or repaired. F. Provide appropriate K3 literature or reference and information for laboratory personnel about change in procedures, methods, technical instructions, and introduction to new tools. H. Monitor sick. G. Develop a schedule of health care activities for laboratory personnel. H. Monitor sick and absent laboratory personnel who may be related to work in the laboratory and report this to laboratory leaders. A. Ensure that used materials and effective waste are disposed of safely after a previous decontamination process. G. Create a call system for emergencies that arise outside of working hours. K. Plan and implement laboratory OSA training for all laboratory staff. L. Record in detail every work sense that occurs in the laboratory and report it to the head of the laboratory.
Laboratory staff head. The executive, the head of laboratory works this must be done. The following: I. Annual cross X-ray for officers. B. Immunization. C. Protection against ultraviolet rays. D. Health monitoring. 3. Facility and infrastructure of G3 laboratory are I. Laboratory code according to standard. B. Gloves. C. masks. D. footwear. 4. Security in case of emergency measures. Need for security in an emergency are I. Heating system. B. Evacuation system. C. First aid kit for a kitchen. 5. Preventive.
Alcohol destruction of lipid structure by penetration into hydrocarbon regions and destruction of protein cell. Short chain alcohol cause more membrane damage than long chain alcohol. Glutaraldehyde to kill bacteria and spora is stronger than formaldehyde. Its activity is able to penetrate the protein layer. Sterilization. Physical method sterilization. Wet sterilization. This method is used to sterilize materials containing liquid or seeds which cannot withstand heat. Conducted with hot steam in a certain pressure. Sterilization often. This method is used to sterilize glassware such as airliner, petri dish, test tube, measuring flask, measuring cups, and others done in the oven. Ethylene oxide gas sterilization used for the sterilization of not heat-resistant materials such as polyethylene tubes, electronic and medical devices, biological agents, and pharmaceuticals. Filtering method sterilization. Filtration is a method of sterilization used for solutions that cannot withstand heat, such as serum, plasma, or urine. Sterilization method of radiation. Ultraviolet irradiation, mainly used to control airborne infection in confined spaces such as tissue culture rooms. Ultraviolet UV rays damage DNA by helping the structure of the cytodimer so that the protein translation process is disrupted. The effectiveness of UV light and deadly substance is closely related to its wavelength. Side effects: damage the retina of the eye and cells that are mitotic so that they are not allowed to work under UV light. In addition, UV rays are mutagenic. Radiation used for the sterilization of hospital equipment in large quantities. The efficiency of sterilization depends on the type of material, temperature, concentration, and resistance of microorganisms to radioactive decontamination of laboratory spaces requires a combination of liquid disinfection and fumigation. The work surface in decontamination beats liquid disinfectant, while fumigation is used for the room and the walls inside. Special measure against blood and body fluids. The actions below are specifically designed to protect laboratory personnel. against bloodborne infections such as hepatitis B virus, HIV, avian influenza, and others. Take label and store specimen. Open the specimen tube and take out the sample. Glass and cloaks blood preparation on a slide automatic equipment.
Specimen management. Receipt of specimen. The laboratory must have a special counter for receiving specimen. If the number of specimen is not large, then specimen assessment can be done a special table in the laboratory. Specimen should be placed in the thickly closed container to prevent specimen spilling. Specimen receiving office. All specimen receiving must wear a laboratory coat. A specimen should be considered infected and held with care. The specimen receipt table should be cleaned with a disinfectant every day. Specimen carrier staff in the laboratory wear a laboratory gown that is strictly closed on the form. When carrying specimen, carry specimen in a special leg tray. If the specimen leaks or spills on the tray is decommissioned and the rest of the specimen is autoclaved. Report to the laboratory staff or quality team if injured during work. Delivery of specimen and infection material from the laboratory. Biology products. Biology products can be finally biology products for humans and animals, made according to regulatory requirements and permits. Final biology product for human and animal research purpose. Biology product for animal tests which are made in accordance with authorities, including unfresh products that have been prepared according to procedure from current government agencies for live vaccines for humans and animals. It is a biological product, not a infectious material. Packaging requires and documentation. Involved material and specimen must be packed in 3 layers from the inside on containing of the watering contains the specimen. The watering container contains sufficient absorbent fat to absorb any leaking specimen fat. The container to place the and contain from extra influence such as special demax and water during the trip. Speaking pack or packing. Delivery of infectious material requires good coordination between the sender, the transport service provider, and the receiving laboratory to ensure that the material is delivered safely and arrives at its destination in good condition.
Waste handling laboratories can be a sort of handheld liquid, solid, and gas waste if not handled properly. Therefore, waste treatment must be carried out properly so as not to cause negative impact. 1. Short, natural, and form of waste. Laboratory waste can come from various sources. A. Expired raw material. B. Consumables. C. Processed product in the laboratory. D. Waste management product. The handling of waste is determined by the nature of waste which is cleaned into. A. Disposal of hazard and toxic material. B. Infective waste. C. Radioactive waste. D. General waste. Each type of waste is disposed of in a spare container, which is labeled according to existing regulation. The form of waste produced can be in the form of a organic solvent, chemical for testing, water from washing tool, remaining specimen, blood and body fluid. B. Solid waste, consumable such as syringes, gloves, cotton swab, specimen bottle, reagent packs, leftover specimen, and culture medium. C. Waste gas, result from using a generator sterilization with ethylene oxide or from a broken thermometer, mercury vapor. 2. Handling and shelter. A. Handling. The principle of waste management is burden and volume reduction. The type of waste must be individual and disaggregated and reduce the overall volume of waste in a sustainable manner. The key to proper disposal into separate hazardous waste differently for all waste in the waste generation set. Place each type of waste in the same bag or container for storage, transportation, and disposal to reduce the possibility of personal and handling error. B. Shelter. It must be noted that the waste storage facility must be adequate, placed in a appropriate, safe, and hygienic place. Complication is an efficient way of storing waste that can be disposed of by landfill. However, complication should not be carried out for infection waste and shop waste. C. Waste separation. To make it easier to identify the various types of waste to be disposed of, use coded bag. However, the use of the codes required sufficient care not cause confusion with other systems that may also use color coding. For example, pocket for ordinary line, salt line, and infected line in hospital and nursing home. D. Standardized waste disposal bag and container.